Okay, I think we are ready. So as Cindy mentioned, my name is Justin Tanner, and I've had the opportunity to work on some Red Blotch projects in Napa County at the Oakville Research Station. And so the topic today that I'll be discussing is results from a couple of our projects looking at different management practices to see what effects they may have on vines with red blotch. So overall, we're going to give you a little introduction into red blotch virus and then speak specifically about the trials that we conducted over the last few years at Oakville. The first one I'll be talking about is the effects of cluster thinning, what effects this practice may have on the fruit quality and the vine physiology, as well as looking at irrigation. And then finally, I'll wrap it up with some kind of summaries of the two trials and talk about, what we can recommend for dealing with red blotch in the vineyard. So first off, red blotch virus was recently identified in 2012. The leaf symptoms are very similar to leaf roll, which probably allowed it to go by for some time, for decades without being detected. However, when many vineyards around Napa were coming up with these red leaves, but testing negative for leaf roll, kind of the flag was raised, and we realized that we're dealing with a new thing. Why red blotch is important for growers is it reduces fruit quality, it can delay ripening, and it's documented across many areas, across the United States, all the way over to New York, and even in wine production regions around the world. And this was probably most likely due to the propagation of vines that were testing virus-free for leaf roll and other viruses. But as I mentioned, just recently discovered that red blotch virus was the causal agent of red blotch disease, which causes these red leaf symptoms very similar to leaf roll. So it's been documented that the virus infects cultivated vitis plants as well as free living vines, both wild and hybrids between Vitis vinifera and wild species along vineyard edges and riparian zones where these vines naturally occur. However, there's been some kind of contradictory reports to what all effects red blotch has in vineyards. There's many different factors when we're looking at the effects of a virus, such as location, weather, different management practices that can kind of convolute the effects we may be seeing in some of these studies. However, some of the reports have mentioned lower net photosynthesis, lower pruning weights, lower vigor. However, reduced translocation likely resulting in the accumulation of carbohydrates in the leaves has been a pretty consistent report in those studies that have looked at this effect, which also makes sense in the reduction of sugars in fruit and the anthocyanins in berries, as less carbohydrates are translocated in red blotch vines. There are less sugars available, less carbohydrates available for sugars in fruit. And then that has a cascading effect into the accumulation of anthocyanins, which are stimulated from sugar accumulation. And sugar actually provides a substrate for their synthesis. So in red grape varieties, this begins around 12 Brix and signifies the beginning of raisin. So speaking a little bit about the leaf symptoms, as I mentioned, very similar to leaf roll. However, there's a couple important distinctions that you can use to kind of tell the difference between the two in the vineyard. For one, both virus symptoms appear on the older leaves at the bases of shoots first, kind of as a reddish or yellow chlorotic, depending on the varieties. In red to black skin varieties, you get this red, which is very noticeable, and you can see in the two photo examples here. However, in white wine grape varieties, the symptom is less distinct and it appears as like a yellowish chlorotic pattern, which first develops in small areas on the leaf, as you can see in this leaf here. However, as the virus symptoms progress, the whole face of the leaf may turn red in both the cases of leaf roll and red blotch. However, with leaf roll, the veins of the leaf remain green, and then there's somewhat of a misshape and kind of cupping or curling or leaf rolling, giving this kind of like rounded shape. Whereas in red blotch, the veins turn red, as you can see in the photo on the right, and the leaf margin remains flat. So there are a couple other symptoms, a couple other things that can cause leaves to turn red, such as certain mineral deficiencies, which usually occur on some of the newer leaves as they're developing and not on the older. And you do not see this chlorotic, this kind of mosaic pattern that you see both with the starts here and there. But early on, before the symptoms really become distinct, kind of later in the season, it could easily be confused with mineral deficiencies. And it's been recommended that if you're facing that kind of situation, you may do a pedial mineral nutrition analysis first before virus testing, because it's cheaper than testing for virus. What we know about the spread of red blotch based off of the distribution of vines over time, it suggests that a flying insect is providing the transmission. And this is a lot to do with like a secondary infection. One of the main ways the virus has been spread initially has been through infected propagation material, specifically before the virus was recognized. And in areas like New York and Switzerland, where they have confirmed the virus is there from propagated material, there's been no secondary spread, which also points to the presence of vectors, specifically in California and Oregon, that is providing a secondary transmission of the virus to new vines. And the three-cornered alfalfa hopper was initially recognized, the first to be recognized as a potential vector. However, it primarily feeds on legumes, as the name implies, alfalfa hopper. Vines are not a great source of food for it. However, as the legumes become poorer quality later in the season, the hoppers may move up into the canopy of the grapevines. However, they're kind of a poor host for the vines and do not allow the three-cornered alfalfa hopper to complete its life cycle. But this insect has been demonstrated to vector red blotch in greenhouse studies. It hasn't been confirmed yet in the field. However, a subsequent study in 2017 showed that it could potentially vector the virus in the vineyard setting. Recently, several other species have also been implicated as potential vectors, so more work is needed on this. And so without really knowing a vector absolutely and the potential for many vectors, it's kind of hard to make recommendations on pest control and spraying for these pests, specifically like the three-cornered alfalfa hopper, as the grapevine is not its main target food and is not present in vineyards to a great degree. So what can we do? Looking into our first project at the effects of crop level and crop thinning on potentially trying to move more photosynthetes from leaves into fruit, could this have an effect in improving fruit quality? It's been shown in healthy vines that this can work. You reduce the crop level, bring the vines into balance, and you can have increased quality. So will this work in red blotch? So for this trial, we looked at 2 years of study, 2020 and 2021. We looked at several different parameters, including plant water status, leaf gas exchange to have a measure of photosynthesis and stomatal conductance. We looked at the non-structural carbohydrates found in leaf and in saps in the second year of study. And then, of course, the components of yield and berry metabolites, both primary and secondary, to look at the effect of fruit quality. So the crop level treatments that we used in this first trial was a full crop load of 45 clusters per vine. We labeled that as the untreated, or a reduced crop level, severely reduced to 10 clusters per vine, in hopes that if this practice would show a beneficial effect, we could then follow up with studies to fine tune the proper level of crop adjustment. However, in going with an extreme example, we were hoping that these effects would be clear off the bat. And so cluster thinning was performed at around 7mm diameter berry size. And we combined the virus status with the 2 cropping levels to have 4 treatment combination groups. Each of these treatment combinations consisted of 4 vines, and the crop level was assigned to vines with and without the virus at random. And then all the cultural conditions were identical in all of these 4 treatment combination groups. They were grown on vertical shoot position trellis system, same cultivar and rootstock. It was Cabernet Sauvignon on 110R, planted in 2012. And the soil type there at the Oakville station is a bell silt loam. So the weather across the 2 years of this study shown in the blue and the red bars. On the left side of this graph, we're showing the growing degree days as they accumulated through the season. And that's shown as the lines that are increasing from April to September. And then precipitation is shown in the bars. So one thing that you'll notice between the blue and the red 2020 and 2021 is significant difference in heat. 2020 was much hotter. So looking at the effect on vine growth through the season, knowing in previous reports that there were differences reported in photosynthetic capacity, where a red blotch may have reduced photosynthesis, we were curious to see what effect would have at a research station in Oakville and its effect on plant water status. So that in the table here, we're looking at the stem water potential integrals. This is the effect over the season from July to August. In the first column, stem water potential, you can notice that there was a significant effect of red blotch infected vines having red blotch, having a slightly lower stem water potential, so a better plant water status. And this effect was seen in both years of study, both in 2020 and in 2021. However, cropping level did not play a significant influence on stem water potential. However, cropping level did affect the stomatal conductance in 2020 and the photosynthetic capacity in 2021, where cluster thin vines had an increased accumulation of carbon in the mesophyll, and an increased stomatoconductance. However, contrary to a few other reports, we see no significant difference with photosynthesis. So both the 2020 year and 2021, they were not statistically different from the uninfected or healthy vines. Then looking at the total soluble sugars in the leaves, we're interested, as this has been reported as something that happens in red blotch vines, is sugars are accumulating in the leaves. And so we wanted to look at what effect the management practice of crop thinning may have at potentially alleviating this issue. So for this, we collected 10 mature leaves from throughout the canopy of each of the vines in this trial. We did this over 3 different dates. We dried the leaves, ground them, weighed them out to 50 milligrams of dried powder, extracted them, and then ran them through HPLC to quantify the total amount of fructose, glucose, sucrose, raffinose, and trehalose. And then what I'm showing here is the total amount of soluble solids from those leaf extracts, and over 3 different points: pre-veraison, at veraison, and mid-ripening. And from these results, we could see that before veraison, there was no significant difference in any of the treatment combinations. However, by veraison and then carried through to mid-veraison, virus status, which is shown by the uppercase letter here, was significantly influenced the amount of sugars in the leaves. So vines that had red blotch retained a greater amount of carbohydrates regardless of cropping level. So we see here the green line, these are the cluster thin vines, as well as the unthin vines. This is a full cropping load with red blotch, both having an elevated level of sugars compared to the red blotch negative vines. So this was kind of discouraging because it shows that regardless of however much we had reduced the cropping load, sugars were still staying elevated, and we're likely not translocating more. However, we wanted to follow this up with looking at carbohydrates that we could extract from shoots. So 1 shoot from each vine was collected at veraison, and we clipped a 7-centimeter segment from the basal node that had a cluster attached to it to see what type of sugars were moving in this shoot. So the 7-centimeter segment was placed in a microtube and centrifuged to extract a little bit of the sap. This would be both phloem and xylem liquid, so it would be slightly diluted from being just pure phloem. However, we still were able to detect a difference in the level of sugars coming in the sap. So it's kind of an inverse situation from the leaf sugars. In sap, we had much more sugar in the red blotch negative vines as opposed to red blotch positive. And so again, the uppercase letter is showing a significant difference in virus status, as the negative vines were significantly higher. However, cluster thinning shown by the lowercase letter did not have a significant difference within these levels. Moving on to yield. In this trial, we can see that red blotch did not significantly reduce yield. This was another contrary finding to a couple other studies in either 2020 or 2021. They were not significantly different. However, when we compared the means from both years together, there was a significant difference where Red Blotch actually had slightly heavier yield. But the big takeaway from this is that adjusting crop level through cluster thinning directly reduces yield. This is the nature of the treatment that we implied. So you can see here in both years, obviously, we cut off the fruit and the result was less yield. However, with Red Blotch, we're very interested in the reduction in fruit quality. So we measured berry weight, Brix, pH, and titratable acidity at 3 times over the season in 2020 and at 4 events in 2021. So this was a veraison, mid-veraison, and harvest, and then pre-veraison, veraison, mid-veraison, and harvest. So looking at berry weight, there was at the beginning of the season, the end of the season, there was no significant difference in 2020. However, there was an effect of virus as shown in difference in uppercase letter here, where Red Blotch significantly increased the berry weight. This is in confirmation of previous studies that have shown that Red Blotch can increase berry weight. And then again in 2021, however, we did see a transient effect kind of earlier in the season at pre-veraison and veraison of cropping level also influencing. So you can see that the cluster thin vines had significantly increased berry weight as opposed to the unthin vines. However, again, by harvest in these 2 years, berry weight was not significantly different. And now looking at pH, we can see at the beginning of the season, at the beginning of veraison and then at pre-veraison in both years, no significant difference. However, cluster thinning did have a significant influence in both mid-ripening and harvest. So you could see that the unthin vines were slightly behind. And then in 2021, same story. We've seen that the unthin vines again were slightly delaying the ripening compared to the cluster thin vines. And then also we noticed a significant interaction of virus and cropping level at the harvest in 2021. Looking at titratable acidity, in 2020, we've seen a significant influence of both virus and cropping level at veraison, mid-veraison. However, by harvest, there was no significant difference. And then a different story in 2021, initially not significant. However, the cropping level was significant at veraison, and then by mid-ripening and harvest, we've seen an effect of virus status. So looking at the fruit quality parameters of anthocyanin content, which is very important for red wine grapes, we see in 2020, there was no significant difference for cropping level and virus status. However, in 2021, we've seen that virus status significantly decreased anthocyanin concentration. These differences may have had to do with the year. 2020 was significantly hotter. These vines are VSP system, which optimizes exposure, ideal for cooler climate situations. And then also the row orientation of these vines was northwest, southeast, which is also a very exposed position. So this may partially explain the no significant differences in 2020. And then flavanols are co-pigment with anthocyanins and also contribute to wine aging. We see no significant differences in either year with either factor. However, the flavanols are very sensitive to UV exposure, and as there was no different canopy manipulation with either of these factors, all the canopy management was held identical. It makes sense that we would not see differences in flavanols. We weren't expecting to see that. So looking at the effect of irrigation, we conducted a 3-year trial where we looked at the same vineyard, same conditions as the cluster thinning project. However, we looked at 3 different irrigation levels. One where we're replacing 100% of the evapotranspiration of the crop, 50% and 25%, to see what effect irrigation may have at improving fruit quality. As we know in healthy vines, that deficit irrigation programs increase the concentration of anthocyanins, improving fruit quality. So we wanted to see, if this will also work in red blotch. So the irrigation treatments that we applied, we used this simple formula. The evapotranspiration of the crop is equal to the observed evapotranspiration from the weather station. In this case, we have CIMMYS Station 77 located on site in Oakville. So very good data directly relating to our site. And then we use the shade cast method from William and Ayers to estimate the canopy size in unstressed sentinel vines within the vineyard, to estimate how much water was lost over a period. In this case, we measured weekly to replace the water that was lost over the previous week to attain these 3 different irrigation levels. So irrigation was supplied by either 2-liter, 4-liter, 8-liter drippers on both sides of the vine. And then we calculated the duration of irrigation based off of the 100% crop evapotranspiration. And then the difference in flow rate allowed us to hit the other replacement targets. So looking at the effect of irrigation on stem water potential, stomatal conductance, and net accumulation of carbon, we've seen that, again, there was a significant influence of red blotch at improving plant water status. So this is a difference of like minus 11.5 bars to minus 10.2 bars in 2020. In 2021, again, we've seen the same trend with vines having red blotch, having slightly less water stress. This overall, this was the integral value, so it's calculated on a biweekly basis from July through August. This is the period of time when we're actually seeing water stress, as we have a Mediterranean climate, and most of the water that we receive through precipitation comes in the winter. So it takes them a little bit of time to dry down in the season. And this is when things really start separating out, this period between July and August. But obviously, irrigation significantly influences the plant water status. And we've seen that consistently in both years. And then we've seen the trend where less water increases the water stress. And then stomatal conductance, we've seen, again, as with the cluster thinning trial, no significant difference in either stomatal conductance or the net accumulation of carbon measured photosynthesis in vines with red blotch. So this was seen in 2020, 2021, and also in 2022. So again, we wanted to see, what effect these treatments might be having on the accumulation of sugars in the leaves. So we performed the same test as we did in the cluster thinning trial, and over the same dates. However, we had 2 years, 2021 and 2022. So looking at the non-structural carbohydrates, this is the total of the fructose, glucose, sucrose, raffinose, and trehalose in these leaf samples. And results are definitely more interesting than the cluster thinning. We're seeing an effect of both virus status as well as irrigation, which is quite interesting. So you can see here that at veraison through mid-ripening in 2021, virus status increased the amount of sugars in the leaves. And then as well as irrigation. So that was both significant in veraison and at mid-veraison. And then in 2020, we've seen the same trend with virus status as well as irrigation initially. However, this effect was dominated by virus status later in the season, but we did observe one significant interaction between virus and irrigation at mid-veraison. So looking at the sugars in sap, over the both years, we've seen in 2020, there was a significant difference. I'm sorry, 2022. However, in 2021, we did not see that relationship. And then in irrigation levels in either year, we did not see a significant difference within the year of non-structural carbohydrates in the sap that we were able to extract from the stem segment. So yield in 2020, we see no significant difference in yield. 2021, same story, and in 2022. So that was quite interesting. However, in irrigated vines, as has been reported previously, without respect to red blotch, less irrigation equals less yield, as a significant portion of the yield is water weight. And so we've seen this trend over the 3 years of study. However, it was only statistically significant in this last year. And then looking at the effects on fruit quality, we looked at berry sampling 3 dates in 2020, and then 4 dates in 2021, 2022. Doing fruit quality for red blotch infected vines. So here you can see both effects of red blotch and irrigation, as more irrigation increases the berry weight as well as red blotch presence. So this trend was seen in each of the 3 years. However, virus status over 2020 and 2021 was significant across the season, but not in 2022. So you can notice earlier in the season, this was at veraison, mid-veraison. We have the effect of vines with red blotch being heavier, and then moving to 2021, we see the same story. However, again, irrigation played a role with heavier irrigation amounts resulting in heavier berries. Same in 2022. So looking at bricks, this is one of the with the reduction in quality for red blotch affected vines is the reduction in sugars that are accumulating in the fruit. So this is a key concern to growers. So again, we can see red blotch significantly reduced. However, the effect was slightly different in the first 2 years as opposed to the last year here, with the difference being in 2022, right before harvest, we hit a really big heat wave, which had a significant role at desiccating some berries, which in turn increases the Brix concentration. And so it was consistent, though, over the 3 years, we've seen effects of virus status reducing the sugar content. And as well, we've seen the trend of irrigation levels increase, seem to reduce that sugar concentration. Looking at pH from veraison to harvest, we've seen initially an effect of irrigation where the less irrigation had higher pH, however, is not significant through the whole season. So by veraison and harvest, not significant. In 2021, no significant differences, and then only a transient effect of virus status in 2022. And then titratable acidity, virus status significantly affected the titratable acidity initially, and then irrigation by the end of 2020 had a significant influence. In 2021, the main effect that we're seeing from mid-veraison to harvest was that virus status at slightly delaying the ripening. And then again in 2022, as well as we're able to observe one significant interaction between virus status right at harvest here. So jumping into anthocyanins as affected by irrigation in red blotch. Over the 3 years, we've seen in 2020 and 2021 significant differences where red blotch positive vines had significantly lower. However, in 2022, no significant difference. And this may be in part to the anthocyanin degradation due to the heat wave, which occurred immediately before harvest. And then effect of irrigation. Here, higher 25% evapotranspiration water replacement has higher anthocyanin concentration across. However, it was only statistically significant in the 2020 and 2022. The trend is present in 2021, but was not significantly different. And as I mentioned, the results in 2022 may be relating to this heat wave that we experienced right before harvest. We harvested this block on September 7th. And as you can see here, we had 5 days above 100 degrees. And this dotted line here represents the 100 degrees Fahrenheit or 37.7 degrees Celsius. And a heat wave is defined as 3 days above this threshold. And experience looking at the relationship in 2022 between the anthocyanins and Brix content in the fruit, we could see that at the 3 time points that we measured the berry quality where anthocyanins were present at veraison, mid-veraison, and harvest, that from veraison to mid-ripening, we see an increase in anthocyanin as well as Brix, whereas more Brix are accumulated, anthocyanins are also accumulating. However, between mid-veraison and veraison, when we had this heat wave event come through, we've seen no statistical difference between the anthocyanin concentration. We've seen significant increase in Brix, as you notice the dots are moving to the right on the x-axis. However, they moved down on the y-axis, showing that the concentration of anthocyanins decreased during this period of time. So some of the takeaway messages from this, looking at the removal of clusters, we did not find a significant influence of being able to redistribute carbohydrates from source to sink. So they were still having elevated carbohydrate level within the leaves of red blotch positive vines. And then in healthy vines, a lower concentration compared to red blotch positive, regardless of cropping level. So the effect of cluster thinning significantly reduces the yield, but did not significantly increase Brix to the same level that we've seen in the healthy vines. With the story of irrigation, definitely more nuanced. Irrigation increases yield. However, lower applied water amounts supports greater accumulation of anthocyanins. However, with red blotch, we're still not overcoming this effect of reduced translocation. And in 2 of the 3 years, we've seen lower Brix and lower anthocyanins, regardless of irrigation status. Like virus dominated the effect. However, in the 3rd year, we've seen increased in Brix that was most likely due to desiccation of the fruit right before harvest due to the heat wave. And so overall, using crop thinning or trying to modify the applied water amounts is not addressing the main effect of the virus is having on the plant physiology, which is a reduction in the translocation of photosynthates from the source to the sink of the fruits. And so as we've shown, we did not observe reduction in photosynthesis with the virus of translocation, which neither of these treatments was able to address, as shown in the level of carbohydrates found in the leaves, in the sap, and then ultimately resulting in lower quality fruit. So we know that we get paid as growers by the yield, and cutting the yield off with cluster thinning does not make financial sense, as it's not improving fruit quality. So the numbers I'm showing here on this table are the amount that we were able to get in 2021 for the fruit with red blotch and without. There is a price penalty. It is significant. However, when you multiply that up by the amount, tons per acre that you can recover, you're losing a huge amount of money and so for that reason, we do not recommend this practice for managing red blotch. It's a very costly approach. However, speaking to a few of the methods we know do work, as vines are infected with viruses, there are no cures. That means once they are infected, they stay infected until they're removed. And so for that reason, starting clean with plant material that's certified to be virus-free when you're starting a new vineyard is the most effective way to, keep the vineyard profitable, as well as staying clean through monitoring and observing for virus symptoms, following that up with testing, keeping track of the amount of symptomatic vines from year to year, their position within the vineyard, one side of the vineyard or the other, if there's clustering, the pattern of spread. And as the years pass and new symptomatic vines develop, it comes to a point where it may make sense to remove the whole vineyard. One study has suggested when more than 30% of an entire block is infected with red blotch, it makes more sense to replace the whole block than to remove individual vines. So with that, I'd like to acknowledge USDA NIFA for funding this project, to the Department of Viticulture and Enology at UC Davis for allowing us to conduct this work in Oakville, to Dr. Khan Kotaral for overseeing this project, designing such an awesome trial and allowing me to conduct this work, to the postdocs that came before me and worked on this trial, Dr. Cliff Yu, and both become assistant professors, to the graduate students that have contributed to making this project possible. It was a lot of data collection, a lot of work. Georgios Pellas helped with this project in 2022, Dimitri Manios in 2021, and Maria Zoom Keller 2021, and Lauren Merigliano both 2021 and 2022. So with that, I would like to thank all of you for your time and attention. This is my email. Feel free to contact me for anything related to red blotch, or if you're in the San Joaquin County or Stanislaus County, I will be moving into the viticulture advisor position starting in January 3rd, and I'm open to helping any way I can with questions related to viticulture production in those areas. And with that, I would like to turn it over to questions.
Thank you, Justin, for your presentation today. I'm going to hand it over to Michelle to ask you. We have a few questions that have come through for you.
Yeah, so the first question, what would be a minimum fallow period to consider if thinking to replant a red blotch vineyard? What cover crops during the fallow period are best?
Yeah, this is a great question. And I'd like to contrast it a little bit with the leaf roll situation. So leaf roll, you can have parts of the vine, like roots and trunk material, still in the vineyard after you remove those vines, that can stay with the virus up to 3 years, it's been shown, as the vector is known. It's the vine mealybug, which can feed on the roots and spread this infection. With red blotch, we don't really understand what the vector is. However, it's not been shown to be translated mechanically on pruners. It is transmitted from infected budwood through grafting, or even grafting onto infected rootstock. It can spread, but there is no, for that reason, there is no minimum fallow period that we know of reported in literature, as opposed to leaf roll. And so with leaf roll, you really need to get out as much of the roots as possible from the field, and then allowing time to pass. It's been shown up to 3 years that it still can transmit. And then with cover crops, that's an interesting question. So I mentioned the 3-corner alfalfa hopper, and how it has a preference for legumes. So I think a great recommendation is made by Dr. Cindy Cron at managing cover crop timing to disrupt the life cycle using the growing degree days. Specifically with the tree hopper, it loves the leguminous plants. So, however, it hasn't been shown that it is the only vector, or even that it's a super efficient vector in the field as well. So definitely there's some questions there, and I wouldn't let that discourage you from using cover crops.
The next question, there are many known plant pathogens that are transmitted by pollen. What has been investigated as a possibility for red blotch transmission? Or excuse me, has this been investigated as a possibility for red blotch transmission?
Yeah, that's a interesting point, but looking at the studies that have looked at the spatial distribution from over time of the red blotch infection, it really seems most fitting to a flying insect vector being what's transmitting it. If it was pollen, it would be heavily influenced by the wind, which is not something that we've seen in these spatial distribution studies. So I think it's pointing away from pollen. I haven't read any reports of looking at that. However, the insect vector is most likely a flying insect vector based off this pattern of infection over time.
Has there been any research on white varietals of red blotch? Or excuse me, white varietals with red blotch? Do you anticipate the results would be comparable?
They would be comparable. However, obviously they're not producing anthocyanin, so you wouldn't see any difference there. However, I think they would. One thing that really these studies have pointed out to me is the difference from year to year, where in this last year, this heat wave really, the vines took a beating during, right before harvest. They went in the healthy vines from like 24 bricks the week before harvest to like 30 bricks, within a week. So definitely sites are influenced differently by weather. In some sites, the symptoms of leaves are really distinct from year to year. Other places, they might not be showing symptoms. And so that the presence of leaf symptoms does not really relate to the effect on fruit quality. You can have effects on fruit quality without ever seeing the symptoms on the vines. So definitely there's other factors that are influencing how much red blotch is affecting from year to year that I'm interested in following up with.
What are your ideas behind why and how GRBV is reducing carbohydrate translocation out of leaves?
Yeah, so I know that Dr. Bashkar has done some amazing scanning electron micrographs of red blotch tissues, and shown there may be something to do with callus formation and buildup that's actually reducing how well the carbohydrates are able to move in the phloem. Showing some like scrambled and distorted looking cells with these weird callus formation, which may provide like a physical impediment to the movement of carbohydrates. And that is kind of congruent with the results that we've seen. No amount of cluster thinning was able to improve the amount of sugars that were loading into fewer fruits, like by channeling more from the source to fewer sinks. And then irrigation, it's kind of a tricky situation because we know that deficit irrigation does improve fruit quality. However, it reduces yield. So there's kind of a balance, yield and quality. However, with red blotch infection and under the 2 normal years, 2021, 20, a significant influence of deficit irrigation improving the anthocyanin concentration.
Do you think it is worth pulling out red blotch infected vines in a young block when you know you have red blotch in another older block nearby? How far do you think red blotch can spread on a particular site?
Yeah, so definitely this is a situation where I would look at the amount of spread between years. If and also the total amount. If by looking at the size of these clusters, that really influences kind of the amount of virus inoculum in the area. If you have a huge block where it's nearly 100% infected and you plant a new block beside it, I would think that the infection level will be much lower in the newer block. And I would focus on, does it make sense to remove the older block and plant a new clean block there? But again, it's going to be site specific where you're going to want to look at the number of vines and their locations that are infected to kind of estimate these clusters and how they may be moving over time. And until we really know, which insects are efficiently vectoring it, I think this is the only way we really can manage is by removing vines, which could be the source for the transmission to healthy vines.
Is there any new research going on? Is there any new research going to be presented this year or early next year regarding new vectors? I know work has been done on scaphy and possible more common leaf hoppers.
Yeah, so definitely I'm not an entomologist. I know that Cindy probably is more aware. Definitely the work is going on. The vectors haven't been confirmed and there's a lot of interest, in being able to manage this. We need to first identify what these vectors are. So definitely, I don't know when new results will be presented or, papers come out definitively confirming vectors within vineyards. So definitely we're all interested in this and are looking out for it.
I can jump on the end of that one. The current research is happening in Kent Dana's lab, and they were looking at scaphy along with other potential vectors, and they do not have any published material, and they were retesting. So that takes a while, sometimes, a couple of years. It's a long process. So I am not aware of them having anything ready to be published anytime soon. But it is still, from my understanding, ongoing research in the Kent Dana lab.
If we can find 3CAH feeding damage, would it make sense to remove the leaf, petiole, or the whole shoot before it spreads to the rest of the vine? How fast does it move through the vine?
Yeah, that is a good question as well. So I know with the initial studies looking at vectors, like in the greenhouse situation, they found that it would take a long time of the insects feeding, like up to 10 days before they would be able to transmit. So definitely not a lot is known. That is something easy to do though, just to pull the leaf off. If you see a leaf hopper feeding there, it's not going to really affect the total photosynthesis, and if you'll have peace of mind. However, we don't know how effective of a method, I guess it really would be.
I can also add a little bit about the virus itself. It's believed the virus moves slowly, but that hasn't been quantified. What does slowly mean? Meaning, how many inches per month does it move? We don't fully understand this virus completely at this point, as researchers in general from all angles. But it is to believe to move slowly, and it is possible. What you're suggesting might help, but there's no research to show that it does at this time.
Is there any literature in the works describing red blotch symptoms on different varieties? I've tested many different varieties. Unfortunately, they test positive, but the symptoms are not exactly the same in Grenache, Petite Sirah, Cab.
Yeah, so definitely the symptoms are different depending on the varieties, where it's most easy to identify the red leaves in red grape varieties. To follow up with that, the site location, I've read, also plays a role, where some sites very regularly show leaf symptoms, where other sites it's hit or miss. In Oakville, we had leaf symptoms that they become most present in the fall, and the expansion of the red areas increases up to leaf fall. So normally in the fall, the leaves in healthy vines go from green to yellow. They don't go to red. So it's not a fall color that we're seeing when we're looking out in the vineyard seeing red leaves. However, in Oakville, I mentioned 2 of the 3 years, in 2020 and 2021, we've seen leaf symptoms. This year, we did not. So they did not turn red for us, and these were following the same vines. So it can, even within the same cultivar, you can see differences in symptoms. So definitely there'll be differences across different cultivars.
Is there any rootstock that cannot be infected with GRRV that can be used next year to replace infected vines?
Yeah, this is an interesting question. We know in leaf roll that there are certain rootstocks that are more sensitive. However, that's not saying that they're not infected. They're saying that they elicit more of a detrimental response with the virus. So as we know of, all Vitis vinifera, hybrids, and even wild species, which is the source of a lot of resistance that they've used to breed new rootstocks with, for having for not being able to be infected. With that said, like, maybe there are differences in the expression, as we've seen with leaf roll. There's certain rootstocks like freedom rootstock, which is sensitive, and coupled with other diseases such as vitivirus, trunk diseases, you can have like sudden vine collapse, which is an extreme response caused by the sensitivity. And with that said, also red blotch itself, there can be more than one clade, and there is more than one clade, and the severity of symptoms may be different as well with the different clades, and as with different rootstocks as well. So to answer directly, no, all the material that we know of can be infected. However, there may be a difference in sensitivity.
Is there any new research out there about how non-symptomatic vines may contribute to virus spread?
Yeah, I have not seen this specific question addressed. However, the non-symptomatic vines are a big issue, right? They're hiding it. They're there, they're present, but they're not showing symptoms, and they provide a reservoir for the virus to be spread by whatever vector may be present, and we have not yet identified it.
Do red blotch infected white wine grapes have a better chance of ripening to an acceptable Brix level than red wine grapes?
So maybe if we look short season versus long season cultivars, with shorter season stuff, obviously they can ripen more, but as far as like when to harvest, this goes back to like wine style, a lot of choices there as to when to pull them. But yeah, I wouldn't say white versus red. I think really the location is going to play a factor, growing degree accumulation, the weather in that season, all of these will influence. In some years, the effect of red blotch is much greater than other years within the same vines, within the same block.
Thank you. We have one more question. Any research on spread efficiency between vigorous vines versus water stressed vines, 3 CAH feeding patterns in those situations?
So I have seen some reports of the three-cornered alfalfa hopper being present more on one side of a vineyard where it was close to like riparian zone, but also I think the feeding patterns mostly going to be related to where their primary food source is. Like I mentioned, the leguminous cover crops, this is where they really want to be eating. However, as the season goes on, the quality of those food sources declines, and that's kind of what forces the three-cornered alfalfa hopper to look for food source elsewhere. And they can feed on the canopy, but it's not their preferred food source. Efficiency to spread. So vigorous vines versus water stress. One interesting thing that we did notice in 2020 and 2021 is a beneficial effect of the virus at improving plant water status, which was kind of curious to us. Like, definitely it was kind of a weird effect. Maybe there's something there like with that, but I don't know, other than like the proximity to riparian zones or to alfalfa fields, I think would play a bigger role on the distribution of three-cornered alfalfa hopper in proximity to the vineyard.